Hi everyone, welcome back to my channel. My name is Jeffrey. In this video, I'm going to discuss with you guys RNAs. Okay, so everything you kind of really need to know about RNAs, and let's just start with the function of RNAs. Okay, what is the function of RNA? And RNA is here to produce protein. Okay, that's basically its entire function. It's to help produce protein. So let's talk about the different types of RNAs. So the first one, you're going to have rRNA. That's going to be your ribosomal RNA. This is going to help make up the actual ribosome itself so that you're able to translate mRNAs into proteins. So obviously, the next step, we're going to talk about mRNAs. So mRNAs, like I said before, are used for protein translation. Okay. And then after mRNAs, you're obviously going to need tRNAs. And the reason why I say you obviously need it is because tRNAs are the ones that actually develop or bring the amino acids. So they actually bring the amino acids so that you're able to form those peptide bonds. So your rRNA is actually able to form those peptide bonds and form a polypeptide for your protein. Because remember, a protein is a polypeptide. And then you have your snRNAs. These are usually called SNRPs or something like that. These are for processing, okay? So this is for example, splicing of your polypeptide, and that forms spliceosomes, okay? So whenever you hear snRNA, just think spliceosome. If I remembered how to spell it, I would spell it correctly. So maybe that's the correct way to spell it, maybe not. Let me know in the comments. So snRNAs, and then you have your final one, which is going to be called miRNAs. These are also microRNAs. These are interfering. Okay, so interfering, I don't know how to spell. But basically this is going to inhibit your mRNA transcription, and it degrades your RNA. So it degrades those transcripts so that your protein is never going to be produced in the first place. Okay. So now we move on and we're like, okay, well, that's great to know all the different types of RNAs. What are the steps to actually make this RNA synthesis? Now, RNA synthesis is going to be from a type of synthesis called de novo synthesis. You might have heard of de novo before, and you might have heard your biology professor actually say something like, oh, DNA cannot de novo synthesize. Essentially, this RNA is just going to be able to be constitutively made, if you will. So you have de novo synthesis, and this constitutes you recognizing your DNA promoter. And after you recognize that DNA promoter, you're going to obviously read it from 5' to 3' because that is how we function. And then you're going to just need some ribonucleotide triphosphates, right? Ribonucleoside triphosphates. So essentially, these triphosphates are going to provide the energy for those bonds so that you're able just to make your RNA de novo synthesis, right? Okay. So there are going to be 3 major types of RNA polymerases. It's going to be RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Okay, so your RNA polymerase 1 is going to be only for rRNAs. Now this is very important. So if you don't, take away anything else, just make sure that you know this at the very minimum. So your RNA polymerase 2 is actually going to be your mRNAs and some snRNAs, as well as all of your microRNAs, okay? And then your RNA polymerase 3 is going to be your tRNAs, along with some snRNAs, all right? So both your RNA polymerase 2 and 3 are going to produce your snRNAs. And remember, RNA polymerase 3 is going to produce that tRNA. RNA polymerase 2 is going to produce that mRNA. And then 1 is obviously the most important. It's going to make your ribosomal RNA, which is going to be your ribosomes. So if you don't have any ribosomes, you obviously can't make any protein. So it doesn't matter if you have mRNAs or tRNAs or not, you won't be able to make protein because you don't have the machinery to be able to do so. All right. So now we move on into transcription termination. Okay, so transcription termination is going to consist of two different things. It's going to have your row independent as well as your row dependent. Okay. Now, what this means is that you just have two different, you just have a protein, it's called row, and one of them is going to not need row, hence row independent, and this is going to be for prokaryotes. Okay, so prokaryotes actually do not have any row protein. It doesn't have to go through row to undergo termination. Essentially, what you're going to have in your RNA is you're going to have a lot of palindromic elements. So in these palindromic regions, when they actually get transcribed, they obviously bind to each other. So if it's GC rich, for example, it'll bind and form 3 hydrogen bonds, which are very strong in comparison to the other bonds. And then this is going to destabilize your DNA polymerase and then disassociate your mRNA from your actual ribosome. All right. So if we had like a ribosome, this is just going to be small subunit, large subunit. You're going to have something that kind of looks like this. You have like a hairpin right here, and then that stalls your RNA polymerase. So eventually, this thing is just going to fall off right here. Okay. And then row dependent is going to have that row protein, right? So you have your row and it attaches to the row recognition site. Okay, so it's going to attach to a recognition site on the RNA, and it moves along. It moves along that RNA. So if we just use the previous analogy here, you're going to have this big row protein here, and it's going to kind of chug along, and then once it reaches that hairpin sequence again, it's actually going to use that helicase activity to just separate the RNA and RNA polymerase from the DNA. Now, additionally to that row-dependent termination, you can also have some things called cleavage factors. So cleavage factors are going to are going to bind to certain sequences in your RNA, and it's actually just going to cleave that RNA. So, for example, you can have these two cleavage factors. It's called CPSF, and then also CSTF. And essentially, these are just going to bind to certain rich elements. So CPSF binds to a AAUAAA polysignal, and your CSTF binds to a GU or U-rich element, and those are going to, it's going to cleave. Now, you don't have to know this for your exam, but it's just kind of interesting, so I'm just going to outline it. Just keep in mind that you don't have to actually know that this is going to be AAUAAA rich, and then the other one is just going to be GU or U rich, okay? So essentially, you're just going to have, CPSF bind right here. You have your CSTF bind right here. And then you have a bunch of cleavage factors, and it's just going to cleave that peptide bond. And you're just going to separate these two strands, okay? Alrighty, so moving on. So now that we've actually terminated our transcription, we have to make some post-transcriptional modifications to our RNA, right? Now, there's going to be two major differences, or one major difference here for prokaryotes and eukaryotes. So for prokaryotes, you're actually just going to say, all right, that's fine. We're just going to directly translate it, okay? And you have 0 modifications, no modifications, all right? And for your eukaryotes, remember, we have all of these organelles in our bodies, and if we just have free rRNA, mRNA just lying around, not bound to anything, we're actually just going to tag it with ubiquitin, and then we're just going to destroy it immediately, right, with the proteasome. So what we need to do is we're going to need to add a 5' cap. For example, you've definitely heard of this, a poly A tail, for example, so that the mRNA actually remains stable, right? So what we're going to have here is we're actually going to have a pretty fancy process that you actually have to know. So the first step is going to have a phosphatase, no, that's an A right there, phosphatase, and that's going to remove one phosphate from your 5'. And after you remove that phosphate, now you can actually add your 5' cap, right? So you're going to have your guanylyl transferase. And transferase obviously is going to transfer or add a 5' GMP to that 5' area where you remove that phosphate from, right? So that's going to be called reverse linkage, just a fancy term for you're adding a 5' cap on, right? And then you are going to methylate it, right? So if you might have heard of this before, you're going to have a 5' methyl cap, right? So you have your guanine 7 methyltransferase, and this is going to add a methyl to the 7 position, okay? And then you are going to have the another methyltransferase. So you're going to have a 2' O methyltransferase. Transferase is going to add a methyl to the 2' oxygen on the ribose sugar. Now, what do you really have to know about from this? I would say know all of the enzymes, just generally, and just know that you need to add that 5' cap, and you need to add a poly A tail, right? So now we're going to talk about the poly A tail, right? So the poly A tail, you are going to have an enzyme called PAP. So PAP is going to recognize the CPSF from before. It's going to add about 200 adenines, so for the poly A tail, and then you have a poly A binding protein, so no PABP, and that's going to bind the actual poly A tail. So you're going to have your poly A tail, and then you're going to have a poly A binding protein on the poly A tail, okay? And then this is going to increase the half-life, right? So poly A binding protein interacts with EIF4G, and it forms a lariat. So essentially what your mRNA is going to look at this point is you're going to have that 5' cap, right? And then you're going to have this poly A tail, and I'm going to just denote this with a ton of circles. But essentially what it does is that this is going to loop around and just form this kind of lariat structure, and that circle structure allows for you to have that increased half-life. And just know that for your poly A tail, it's going to have your poly A binding protein, and that poly A binding protein is going to interact with EIF4G to stabilize the lariat structure, okay? So this is a very important concept. I think this is one of the most important concepts because it's guaranteed to be actually tested, okay? So, yeah, cool. And then you also have this thing called splicing. Splicing is going to be in eukaryotes. And basically splicing, you're going to have an exon, right? And then you're going to have an intron. And then you're going to have another exon. And remember that exons are important, right? So exons are what you need to make the protein that you currently want to make. Introns are what needs to be removed, right? So for splicing, you're essentially just exiting out this intron. You're going to take out that intron, and you're essentially there's going to be a ton of things that you learned. You learned about the different 5 SNRMPs, the SNRPs, and these are U1 through U6, and or minus U3, and they form a lariat complex, and they attach onto branch point A, and they splice the exons together. They form a lariat structure, and then that lariat structure actually just falls off because of the spliceosome and all of that good stuff. And you really just have to know that there's a SNRMP, right? So you don't have to know the U1, U2, U3, U4, U5, U6. Like you don't have to know any of it. Clearly, I forgot some of it too. So just know that you have SNRMPs. They form a lariat structure on branch point A. So they form lariat on branch point A. You don't have to know which A. You don't have to know anything about that A. Just know that it's in the middle of the actual intron. So that A is going to be somewhere here. And this RNA is going to loop around. It's going to form that lariat structure. And then you're going to have the other spliceosome. So the entire spliceosome is covering this. And then that's just going to take this part of the exon, bind it to this part of the exon, and then that's going to be splicing. All right. So you don't have to know the U1. Just know that like those things exist and they help you splice. All right. And then you have alternative splicing, for example. So these are going to be the same gene. So you're going to have this gene right here, and you're just going to, so for example, alpha tropomyosin, and some people might just splice out this part, make a protein, but then some other people, they might actually, start splicing here and end up splicing here, and they only have one splice spot, and it forms, even though it forms the same protein, technically, functionally speaking, it's the same protein, but it's a different amino acid sequence, right? Because you're splicing it differently. So you're obviously going to have some sort of difference. Now let's move on to what is even more important in my opinion, which are the clinical correlates. All right, so make sure that you know your drugs. I mean, this is pretty much given. There's going to be a lot of drugs that you learn, and I'm going to actually link down in the comments below my own drug list that I made. So it includes all the drugs that you need to know for this first exam. But basically, you have to know what AZT does. You have to know what DDI does. You have to know what tenofovir does, cytosine, arabinoside, right? I call that Arac-C. You have to know what etoposide does, actinomycin D, rifamycin, all these different drugs. Make sure you memorize them all. I included it, so just print out a copy, make some notes on it, and memorize it all. But yeah, so just know all the drugs. There's going to be 2 deaminase reactions that are very important. So you have your adenosine to inosine, so your A to I, and then you also have your cytidine to your uracil, so your C to your U. So these are, the enzymes are very self-explanatory. It's adenosine deaminase, and then also cytidine deaminase, right? Very self-explanatory enzymes. So for your adenosine deaminase, it obviously does a different reaction than your cytidine deaminase, but your adenosine deaminase is essentially just going to go from a glutamine to a arginine, okay? So what that does is that this is supposed to be normal, and obviously undergoing this deaminase reaction, that's going to be making it abnormal. So essentially this is going to affect your glutamate channel, And your glutamate channel typically conducts both your sodium and your calcium. So calcium, right? But now what happens is that because it's an arginine, it's going to make this glutamate channel only conduct sodium, okay? Which is a big problem. And then your cytidine deaminase is what is going to create a stop codon. So it's just going to make a stop codon. Obviously, that makes a lot of problems because this is going to terminate apolipoprotein B. So I don't want to spell it out, but apolipoprotein B, and this is going to be pathological in your intestine. Right? Okay, so your apolipoprotein B is going to be messed up and it's going to be in your intestine. Now you also have 3 different diseases that you have to know about. So you have your systemic lupus erythematosus. So that's just going to have your antibodies attack your U1. So if you remember from when I talked about U1, which is very briefly about the SNRPs, that's going to affect your spliceosome, right? So antibodies to U1. And this is going to affect your U1, your histones, as well as your topoisomerases, okay? Isomerases, okay. Okay, so moving on now to beta thalassemia. Beta thalassemia is going to be a mutation in the normal acceptor site. And this is going to skip for, so your SNRPs, which is your SNRMPs, are going to find the next acceptor site to splice. So just to give you guys an example of what I'm talking about here, you're going to have your, let's just say exon 1 here, right? Exon 1, you're going to have your intron 1 here, you're going to have your exon 2 here, and you're going to have your intron 2 here, and then exon 3, okay? So what's going to happen is that you normally have an acceptor site at the end of the intron, right? So this is where you're going to cut out that intron, and then you're going to splice together these 2 exons, right? So that makes for a normal protein. Essentially, this intron, this acceptor site is defective, so that means that your SNRMPs, remember these actually do help with the splicing, they're not going to recognize that acceptor site. So they're going to keep on moving until the next acceptor site. So they are not going to actually cut at that normal acceptor site. They're going to cut at the next. So what happens is that now you're cutting out essentially intron 1, you're cutting out intron 2, right? But you're also cutting out exon 2. So that's a huge problem because now you have a shorter protein, right? You don't have that second exon. So essentially you're just cutting out 2 introns, which is what you want, but then you're cutting out an entire exon. So that's going to change your reading frame. It's going to make for maybe a non-functional or a dysfunctional protein. So that's a big problem, all right? And then you have your limb-girdle muscular dystrophy. Limb-girdle, essentially what this is that it's a mutation in the exon, and that causes a strong splice site or the acceptor in the middle of a codon. So this is going to cause a frame shift. So if we're just going to talk about what we were just talking about before, so we have your exon 1, your intron 1, your exon 2 here. Essentially what happens is that you're going to have a mutation and it's going to happen in your exon, right? So let's just say that happens in your exon 1, right? So you're going to have a mutation that's going to cause a very strong acceptor site, and that is going to actually allow for splicing, right? So you're going to have a SNRMP, you're going to have a SNRP, it recognizes it, so it's going to excise out this entire exon. So that's just going to cause a frame shift mutation, and that's why limb-girdle is a very serious medical condition. Now, I hope that this video has been helpful. I've been a little over the place, but I hope that you still took something out of it and that it was helpful for your education. And have a great day.